Thank you all, and thank you to Team Phenomenal Hope for the chance to speak today. My name is Vineet Agrawal. I work in the laboratory of Anna Hemnes, and I wanted to show you some of the progress we've made over the last year with your generous support. The title of my talk today is A Pathogenic Role for the Natriuretic Peptide Clearance Receptor in Pulmonary Hypertension. There we go. So today's talk isn't really meant to inundate you with data, but hopefully give you some broad strokes and a general overview of what we've been working on, why it's important, and how we're moving forward with your support. Specifically, I'll tell you a little bit about pulmonary hypertension due to heart failure with preserved ejection fraction, or HFpEF, and actually we've heard a good overview of it from Dr. Lai recently. And I'll tell you a little bit more about the target that we are looking at, the natriuretic peptide clearance receptor, in both an animal model and how we're beginning to study it in humans as well. So in the interest of time, I'll gloss over this since Dr. Lai did such a great job of explaining what group 2 pulmonary hypertension is, or pulmonary hypertension due to left-sided heart disease, and why it's such a problem. You know, as she mentioned, it's the most common form of pulmonary hypertension, and amongst the various forms of pulmonary hypertension currently has no treatments. And amongst those different groups, amongst the various causes of pulmonary hypertension due to left heart disease, they can be broken down into valve problems. So either valves that are too tight or too leaky, or it can be a pump problem, the pump not actually pumping well enough, or the pump being too stiff and causing backflow, as Dr. Lai had shown you quite nicely. Amongst those various different forms of group 2 pulmonary hypertension, really there's only one that doesn't have any treatment options at this point, and it is pulmonary hypertension due to HFpEF, or the stiff heart that leads to high pressures in the lungs. And so, this was really the motivation for me to try and understand the underlying disease processes that lead to pulmonary hypertension with HFpEF. The reason why we don't have treatments at this point is in large part because we don't understand what causes it. And you know, that process requires not only preclinical animal models, but also picking the right one, as Dr. Lai had also mentioned. You know, there are many models in the literature, many of which are in some ways imperfect. We picked a model that we think matched what we think we see in patients. You know, that there's a growing evidence for the role of obesity, diabetes, high blood pressure in both the development of pulmonary hypertension and HFpEF. And actually using a model that Dr. Lai and one of her colleagues at the University of Pittsburgh had initially published in the literature, we found we used the same model where we fed the animals a high-fat diet, and they spontaneously develop pulmonary hypertension and HFpEF. And using that model, we wanted to look at early changes that occur that could potentially lead us towards an interesting therapy. And so I won't go through all of the data that we collected over the last year, but I do wanted to share a few pieces of data with you to show that the model we picked, we think actually does mimic aspects of the human disease. And so on the left side of this slide here are a group of pictures that show in the top left, and hopefully you all can see my mouse, that the animals that we gave a high-fat or Western diet actually do develop increased pressures. And these pressures were measured using very small catheters that we would use, similar to the right heart catheters or the Swan catheters that you would have placed in your neck. We use similar type catheters in animals to directly measure the pressures. To confirm that these pressures were actually high, we also looked directly at the lungs. And similarly, the mice that we gave the high-fat diet to developed these thickened vessels around, thickening around the vessels in the lungs, whereas the ones who don't get it don't. And again, sort of mimicking what we see in humans, when we used a special technique called staining to look at what proteins are expressed in those thickened vessels, we found that a number of proteins that we would expect to see in humans who have the disease were also increased in this model. And one of which is alpha smooth muscle actin, which you can see in smooth muscle cells and sometimes in the inner lining of vessels or endothelial cells when they start to undergo changes. On the right side of this slide, I wanted to just show you what the hearts of these mice look like when they are given a Western diet. And these are cross sections of the heart where you can see on the left side is what we call the left ventricle, somewhat of a donut shape here. And on the right side in this moon shape or this crescent shape is the right ventricle. Hopefully I can convince you that the mice that develop pulmonary hypertension and HFpEF have larger hearts, larger left and larger right-sided hearts. So after confirming that we thought we had a pretty good model of pulmonary hypertension with HFpEF, we wanted to look at what changes occur that lead to pulmonary hypertension and HFpEF with the idea that perhaps we can find a therapeutic target. The way we did this was we wanted to look specifically at changes in the expression of RNA in the various tissues, the heart and the lung. RNA is one of those is a molecule within cells that essentially forms the blueprint of proteins that then form the building block of cells. And so by looking at RNA, you can get a sense of what changes are happening at the cellular level. So after looking at over 13,000 RNA transcripts, we noted that the most differentially changed transcript encoded this protein called the natriuretic peptide clearance receptor. Well, what is this receptor? Well, as some of you may recall, the acronym BNP, I've been at a clinic visit. It's often used in a clinical setting to measure how much stress the heart is under. BNP is one of a family of natriuretic peptides that are released by various parts of the heart when the heart is under stress. That stress can be due to pulmonary hypertension, due to the heart itself, or due to a number of other potential issues. And when these natriuretic peptides are released, what they do is they bind to these receptors on cells that upon binding allow a number of changes to occur in the cell that can, in many cases, if they bind the right receptors, decrease pressure in the lungs and in the rest of the body, relax the heart, and prevent the heart from overgrowing too much. 2 out of the 3 receptors that the natriuretic peptides bind actually have those beneficial effects. The third one, which is the clearance receptor, actually serves as a decoy receptor. What it does is it binds to these same natriuretic peptides, but rather than allowing the changes to occur in the cell, it just internalizes it and degrades away the natriuretic peptides so that they can't actually do the good things that we would want them to do. So to confirm that what we were looking at could potentially is actually occurring in our model, we started to develop cells where we could artificially increase the expression of this clearance receptor. And so in the bottom left here, I have a specific type of cell, a cardiomyocyte-like cell, like a heart cell, where what we did was we artificially increased the expression of this clearance receptor. And hopefully I can convince you that these cells went from looking quite small to quite large. And the stain that we used here is supposed to outline the fibers within the cell. And what were fibers normally align in a heart cell, fibers should normally align in one direction. And you can see in these larger cells, they start to form what we call disarray or align in various different directions. So I showed you so far that the clearance receptor, if it's increased in cells or potentially in animals, can cause pulmonary hypertension and HFpEF-like features. Well, how do we know for sure? The best way to know is to try and fix the problem by targeting the same receptor. And so what we did was, using the same animal model, we then treated these animals with a drug that specifically targets this clearance receptor. And this work is still ongoing, but I wanted to show you some of the preliminary findings. In the mice that receive this targeted drug, on the left you have normal mice. In the middle you have the mice that develop pulmonary hypertension with HFpEF. And then on the right of each of these graphs, you have the mice that were given the drug. We can actually prevent the increase in the pressure within the right ventricle, which we use as the surrogate for pulmonary pressure. And also by ultrasound, we were able to show that the increased thickness of the right ventricle that we saw in our mice actually was prevented as well. So we think that this is all very suggestive that the clearance receptor may play an important role in the development of pulmonary hypertension. So that was a little bit of a whirlwind tour of what we've been doing to study pulmonary hypertension and HFpEF in mice over the last year. But what about humans? So it turns out, as you all may have surmised, mice are not humans. And many therapies that we find in mice don't always work in humans. You know, the gold standard way of knowing is to run a clinical trial, and we heard some very fantastic talks about ongoing clinical trials. Getting to that point can be time consuming, requires oftentimes a large number of patients. It can be expensive. And especially if the end result is negative, it's a lot of time and effort that, could have, it sometimes feels as though it could have, it was a large use of resources that could have, it feels a little bit like it was wasted in some ways. Not that that's always the case, but the question always arises, is there an intermediate or alternative solution that could potentially give us more information? And that's something that we've been working on over the last year as well. I wanted to share some of that exciting direction with you. So to look at an alternative method by which we could study humans and maybe even look at the biology of humans without having to necessarily conduct a large clinical trial, I'm going to tell you about what I would call a virtual clinical trial. And this approach requires two things: the presence of a large database of patients with clinical diagnoses and information, and some information about the DNA sequences that each of these patients harbors. It turns out that the institution I work at has a great resource for this called BioView. Since the 1990s, Vanderbilt has taken every patient who consents to this process and transferred their entire medical record full of diagnoses, lab results, medications, and found a way to scrub the electronic health record of any identifiable information so that it can never be traced back to that same person, and put it into a research database that has all the information we would be interested in as clinicians and as researchers. They also would take any leftover blood that would have been sent off to the lab for testing, not all of which is always used, and using the leftover blood would isolate DNA from that blood. So now you have a database where there's information about patients, unidentified, linked to DNA. But why is the DNA important? Well, that's where the virtual clinical trial idea comes in. All of us are humans and have about 99% of our DNA is fairly identical. But as you know, we are not all identical. And it's really that 1% difference between all of us that gives rise to all the different changes between all of us. And within our DNA, which is encoded by 4 different bases, A, C, G, and T, in various different configurations, our DNA uses those 4 bases to give rise to all of the proteins and the cells and the tissues in our body. And every so often in our DNA, there are segments that vary from one person to another. When a single base differs, we call this a single nucleotide polymorphism, or a SNP, as you can see. In the right regions of the DNA, these SNPs can actually affect the expression of proteins. And so here you have an example of 3 different people who have in a single position 3 different bases. And in the right region, in one patient that can cause an increase in expression of a receptor. In another region, it can cause no change. And in a third patient, sorry, it can cause a decrease. You can use that information by knowing the effect of any given SNP on the expression of a protein to conduct what I've called a virtual clinical trial. Because in essence, what drugs do is they can either decrease the effect of a target, increase the effect of a target, or so on and so forth. And so by knowing that certain genotypes are associated with lower or higher expression, and then looking at whether those genotypes actually in a large database can affect the right ventricle or pulmonary hypertension, you can make some inferences about the likelihood of a therapy being effective. And also the likelihood that a target like the clearance receptor has an important role in the biology of the disease you're looking at. It also turns out that in studies that have shown a genetic link, as I've alluded to, between a target and a disease, drugs that target that same target are 2-fold more likely to actually get FDA approved down the line, suggesting that there is some validity to this approach. In the last few minutes, I'm just going to show you some of the preliminary data we have with this approach. We're looking at a number of different variants that affect this clearance receptor, but one that's particularly interesting, in part because it's common, and in part because we think it has a pretty potent role in affecting the expression of this clearance receptor. In patients who harbor this one variant, which is a long name, RS227095, if they harbor 1 or 2 copies of this variant, in our large database of over 6,000 people, there's a statistically increased likelihood that they have a slightly higher pulmonary pressure. Now, this is estimated by ultrasound, so it's not the gold standard right heart catheterization, but nonetheless, we think it's suggestive. And given what we know about this clearance receptor's role, we also looked at whether the presence of this variant affects the amount of natriuretic peptide that's out in the blood. And the more of these more copies of these variants that a patient possesses, the lower the BNP levels are in the blood. And finally, when we looked at the measurement of right ventricular mass by cardiac MRI, the same thing was seen, that the more copies of this variant that were present, the greater decrease in RV mass, all suggestive that this clearance receptor may actually also be important in humans. And so, in conclusion, hopefully I've convinced you that the natriuretic peptide clearance receptor appears to be a functional target in pulmonary hypertension and HFpEF in both animals and humans. We've still got quite a bit of work to do to understand the why, the biology underlying why this clearance receptor may affect pulmonary vessels and the right heart. And we're certainly working on that as we speak. And I just wanted to finish off by thanking Team Phenomenal Hope for their support. Not only did this allow me to do the work that I was able to do in tackling the problem of pulmonary hypertension and HFpEF, but I think most importantly, it served as validation that what I do, to get up every morning and go into the lab matters to the very people who have pulmonary hypertension and take care of patients with pulmonary hypertension. I think that is probably the most important thing that I've taken away from this support and opportunity. With that, I'll stop and take any questions. I'll say that it takes a village, and I'm the one who got to get up here and talk in front of you all today, but the credit goes to many other people. In the top right is a picture of our pulmonary vascular biology group at Vanderbilt, a large group of folks who work every day to try and find solutions to pulmonary hypertension. Thank you.
Fantastic. Thank you so much. It's incredible when our research committee, review committee met about this last year and saw there is, as explained in your talk and the talk prior by Dr. Lai, there's such a dearth of knowledge in PH-HFpEF. And we see these patients a lot in our clinic. And so without effective treatments. So thank you so much for doing this research and doing this work. And, congratulations on what you shared with us today.